Hi, I'm Sue Biggins, and I'm an investigator at the Fred Hutchinson Cancer Research Center in the Division of Basic Sciences, and I'm also an investigator with the Howard Hughes Medical Institute. And for part 2 of my talk, I'm going to discuss our work investigating kinetochore function. Just to remind you, chromosome segregation is mediated by kinetochore-microtubule attachments. At metaphase, microtubules emanating from spindle poles attach to the chromosomes through structures called kinetochores that I discussed in detail in the first part of my talk. Once every pair of chromosomes is properly aligned, the cell enters anaphase and the microtubules physically pull chromosomes to opposite poles. Now, as I described in the first part of my talk, kinetochores carry out really complex functions. They have a mechanical role where the kinetochore has to stay attached to the dynamic tip of a microtubule that's constantly growing and shrinking from under it. They have interesting biophysical properties where tension stabilizes attachments. And they serve as signaling hubs where a single kinetochore can completely halt the cell cycle if there's a defect in kinetochore-microtubule attachments. Now, to try to understand more about how kinetochores work, it turns out there's a number of challenges to studying them. First, kinetochores contain hundreds of proteins, and this has made it difficult to understand what the individual contributions of proteins are to all of these functions, especially because often when you mutate one, a lot of other kinetochore proteins are no longer localized to the kinetochore. Attachment and tension are completely coupled in vivo. As soon as proper attachments are made, they quickly come under tension, and that's made it difficult to understand the contributions of attachment and tension. We can't manipulate tension in vivo, so it's really difficult to directly probe and put kinetochores under tension in cells to ask how that affects their properties. So many years ago, I decided that to really understand some of the functions of kinetochores, we should try to reconstitute kinetochore-microtubule interactions to do direct studies in vitro where we could probe them and put them under tension and ask how they behave. Before I tell you how we did that, I want to introduce you to the model organism we've been using for these studies, which is the budding yeast kinetochore. This is just a schematic cartoon of what the budding yeast kinetochore looks like. It's the simplest kinetochore. There's just a single microtubule binding site that binds to a single centromeric nucleosome in the kinetochore. The inner kinetochore contains the proteins that directly bind to that centromere, and then the outer kinetochore are the proteins and complexes that directly mediate attachment to the microtubule. So multiple subcomplexes link the chromosome to the microtubule, and these subcomplexes are present in multiple copies, as I mentioned in my first talk. Now, despite the relative simplicity of the budding yeast kinetochore, the bulk of the functions and subcomplexes are conserved. The major microtubule binding activity in the yeast kinetochore has been attributed to two complexes called NDC80 and DAM1. And now I've just simplified the cartoon, and I'm just showing a single DAM1 complex and two NDC80 complexes. In green is the NDC80 complex. This is a 4-component complex, and there's about 8 of them in a single yeast kinetochore. And mutants in vivo have the most severe attachment defects, which is one of the reasons this has been considered to be the major microtubule binding component of the kinetochore. Now, the DAM1 complex is shown as this ring here. It's a 10-component complex, and we think there's about 16 of them in a kinetochore that can form a ring around the microtubule, and that was nicely shown by the Nogales and Harrison labs. And mutants in vivo are defective in end-on attachments. So in thinking about how to reconstitute kinetochores, the question is, how can we obtain kinetochores to work with? And there are two major approaches. One would be to reconstitute them using recombinant subcomplexes. So the idea here would be to make each of those subcomplexes in bacteria or another organism and then try to put them together. And the other obvious approach was we could try to purify native kinetochores from cells. And there are issues with both of these approaches. First, the complexity of the kinetochore. As I said, it contains many, many subcomplexes, and even in budding yeast, there's at least 9 subcomplexes present in multiple copies. So it's really complex. The template, as I described in the first part of my talk, the kinetochore assembles on a specialized chromatin structure, and at the time, no one had reconstituted that chromatin structure, so we didn't know if we really could make the template for the kinetochore to assemble. And finally, the regulation. There's really not much known about any sort of post-translational regulation or cell cycle requirements for the assembly process. So at the time, reconstituting the kinetochore seemed like a big challenge and very difficult. Now, there were also challenges to purifying native kinetochores. First, purity. As I mentioned, the kinetochore is on the chromosome, and it's just a single locus on the chromosome. So we didn't know if we could come up with a purification strategy that would be clean enough and not have contaminants from other chromosomal proteins. Abundance was a huge issue. One kinetochore on a chromosome. We're completely limited by the number of chromosomes in a cell. And then finally, the size. A single even yeast kinetochore is estimated to be greater than 5 megadaltons, which is bigger than a ribosome. And so we really didn't know if we could come up with a purification strategy that would maintain such a large complex in its native form. So in thinking about these things, we decided to take the approach of trying to purify kinetochores. And what I'm going to tell you today is we were able to achieve that. Bungo Akiyoshi, a really talented grad student in my lab, developed a purification technique. So it turns out, this is again a schematic of the yeast kinetochore, that he flag epitope tagged one component of a complex called MIS12. This is a 4-component subcomplex. When he epitope tagged the DSN1 protein and purified it, he developed conditions where he could purify most of the kinetochore. And that's shown on this silver stain here. On the left is a negative control, and on the right is the DSN1 flag purification. And you can see that he purified many more than just the 4 components of this subcomplex when he purified DSN1. So we did mass spec to determine what those proteins were in that preparation, and it turns out that mass spec identified greater than 95% of kinetochore proteins. So we had proteins from every single kinetochore subcomplex with the exception of the CBF3 complex, which is a yeast-specific DNA binding complex. So we had achieved purification of most of the kinetochore in this single step. And on that silver stain gel I showed you earlier, almost all of those proteins are kinetochore proteins, and Bungo showed that by epitope tagging them and showing that when he repurified the kinetochores, that the epitope tag protein would then move on the silver stain gel. There's also substoichiometric proteins that we can see by immunoblotting. We also have regulatory and spindle checkpoint proteins present in this prep. However, it turns out we have no Aurora B or motor proteins. And there's no Aurora B because in budding yeast, CBF3 is the receptor for the Aurora B kinase. We then performed gel filtration to see if all those kinetochore proteins were really present in a single complex. This is fractions from a gel filtration column, and these are just some representative fractions. And then what we did is immunoblot with representative antibodies spanning the inner to the outer kinetochore. And you can see that we have components throughout the whole kinetochore present in a few fractions. And that told us that we have at least some full kinetochores in our preps. Okay, so now once we had purified these things, the next big question is, well, are they functional? Can they do any of the functions that kinetochores do in the cell outside of the cell? The first question is, can they just bind to a microtubule? So kinetochores initially just bind anywhere on the lattice of a microtubule, and that was our first question. Can they stay bound to a growing and shrinking tip? If you remember from my first part of my talk, the kinetochore has to maintain an attachment to a dynamic tip, and so that was the second question. And then they have to be able to form load-bearing attachments where they come under tension, because kinetochores come under tension in the cell. And so that was our next question. We set up a really great collaboration with Chip Asbury's lab. He's a biophysicist at the University of Washington. And with his lab, we took biophysical approaches to address these 3 questions. And I'll tell you what we did. First, we made the kinetochores fluorescent so that we could see them under the microscope. And so you can GFP tag any kinetochore protein, but for the work I'll show you, we GFP tagged the centromeric histone called SEMP-A. We then purified those kinetochores so that they were fluorescent green due to the presence of SEMP-A. We then made microtubules, and this is easy to do in vitro with purified tubulin. And we treated them with Taxol, which is a drug that stabilizes the microtubules, so these are not dynamic. And then we used rhodamine as a fluorescent tag to make them fluorescent so we could see them under the microscope. So we attached rhodamine-labeled microtubules to a cover slip, and then we just flowed the purified kinetochores in and asked, can they bind to the microtubule? So this is an image of our work, and you can see in red are the microtubules, and in green are the kinetochores, and you can see that the kinetochores can bind all over the microtubules. So this confirmed that the kinetochores we purified are functional to bind to microtubules. Bungo went through and showed that the association depends on the microtubule binding proteins in the kinetochore, such as NDC80. And he did that by just purifying kinetochores from various mutants, where then they were lacking that complex. Okay, so the next question then is, well, can they maintain an attachment to a dynamic tip? And to do that, we developed the following assay. We take a microtubule seed and we attach it to a cover slip. We then grow a dynamic microtubule off of the cover slip, and we do this in the presence of the kinetochores so that they bind all along the lattice of the microtubule. Then, to induce microtubule disassembly, we flow out the free tubulin, and that causes the tips of the microtubules to disassemble. And I'm going to show you this in the next slide in a movie. So what you're looking at here is the microtubule, and you can see a number of kinetochores bound to the side of the microtubule. And when I start the movie, if you watch the bottom of the microtubule, when it starts to disassemble, you'll see that it hits that first green kinetochore, and that kinetochore stays attached. So I'll start the movie for you. So you can see now the microtubule is disassembling, and the kinetochore is staying attached to that microtubule as it disassembles right out from under it. So that showed that our kinetochore can bind to a microtubule tip. And I want to emphasize, as I said earlier, we have no motor proteins in these preps that could drive kinetochore movement. So what's happening here is that the ability of the kinetochore to stay bound just to the microtubule tip is sufficient to have it move the kinetochore. Next, we wanted to ask, can we put these under tension? And to do this, we developed yet another assay, and this is a laser trapping or optical trapping assay. And what we do here is the setup is exactly the same. We grow a dynamic microtubule off of a cover slip, but now what we do is we take large polystyrene beads and we attach kinetochores to those beads. And so the beads serve as cargo that we can hold. They sort of take the place of the chromosome. We use a laser trap to capture that bead, and then we can position it at the tip of a microtubule so that a kinetochore binds to the tip of the microtubule. And then we can do different things. We can apply tension using a feedback-controlled stage. We can vary the tension, or we can keep the tension constant. So using this assay, we were able to analyze a kinetochore attachment to a microtubule tip under tension. So what you're seeing here is a trace of a single experiment where we attached a kinetochore to the tip of a microtubule. And in this experiment, we maintain the tension at 1.7 piconewtons. And in these experiments, we're filming them so we can see what's happening to the bead and the microtubule. And on the left, if you look at the bead position, the relative bead position tells us what's happening to the microtubule. So if the bead is moving in one direction, we can see that the microtubule is assembling. And then you can see it suddenly switches to moving in a different direction. And that's the microtubule switches into a state of disassembly. And then if it switches between these phases, it's either a rescue or a catastrophe event. At some point in the experiment, the kinetochore will just let go. It'll detach from the tip of the microtubule, and at that point, we call that the total lifetime of the attachment. So that's the length of time that the kinetochore stayed attached to that dynamic tip. And so you can see in this experiment that we learned a lot in this single experiment. The kinetochores can stay bound to the tip of a microtubule under tension, and we found that these particles can remain bound through all the dynamic phases that microtubules undergo. So at this point, we were really convinced that we had reconstituted dynamic kinetochore-microtubule attachments under tension. These purified kinetochores maintain attachments much better than any individual subcomplex has been seen to maintain an attachment. And so in the past, work from various labs have reconstituted attachments between the NDC80 complex and the NDC80 and DAM1 complexes to the microtubules. And that's shown here. This is work from Chip Asbury's lab and Tricia Davis. And you can see that with the NDC80 complex, if they coat beads with NDC80 and then put it on the tip of a microtubule, what you're looking at here is the probability that an attachment will survive a given time. And so when they add DAM1 to NDC80 beads, it definitely improves the performance. But if you look at our kinetochores, they greatly outperform either of these single microtubule binding complexes. And I don't have time to go through it, but we were able to show that in our experiments, single kinetochores suffice for this coupling activity, whereas in the experiments with NDC80 and DAM1, multiple subcomplexes were involved in the attachment. So the kinetochores, native kinetochores, really do outperform any individual microtubule binding complex that's been seen before. So having purified kinetochores, then, let us really address these functions that I talked about in the first talk. And so today I'm going to tell you some of the work we did to look at the mechanical role of the kinetochore binding to the microtubule, and then we'll talk about the biophysical properties. So as I discussed in the first part of the talk, there's various models for how the kinetochore might maintain an attachment. Now, the original model was that motor proteins at the kinetochore could maintain an attachment and walk along the microtubule. And this was actually the most popular model until we found out that motor proteins are actually not essential for chromosome movement in cells. So they're extremely important, but they're not driving kinetochore attachment and movement on the microtubule. There's ring models that take into account the idea that the kinetochore can form a ring around the microtubule, and that as long as that can stay attached, the peeling protofilaments as the microtubule disassembles will maintain an attachment. They'll harness the energy of depolymerizing microtubules. And this model has been very popular because work from the Nogales, Barnes, and Harrison labs, as you can see in these EM images, showed that the DAM1 complex can form a ring around the microtubule. Biased diffusion models are also very popular, and the idea here is that multiple microtubule binding elements within the kinetochore rapidly bind and unbind to the tip of the microtubule, and as long as this occurs in a biased manner, it would maintain an attachment near the tip. And this model, these are just EM images of the NDC80 complex to show you that they have, there are long coil proteins that have globular domains that bind to the microtubule head, and the Asbury and Davis labs showed that this complex can undergo biased diffusion. So we still don't know what the actual underlying mechanism is. And part of the difficulty has been that in vivo, it's very difficult to visualize the structure of a kinetochore-microtubule attachment. So this is an EM image from Bruce McEwen's lab, and you can see the underlying kinetochore structure, and then you can see microtubules attaching to it. And it's very difficult to look at this structure and really understand how it's attaching. So we thought perhaps if we did electron microscopy on our purified kinetochores attached to microtubules, we might learn more about the structure. We set up a collaboration with Tamir Gonen's lab, and we were able to see in EM, in negative stain EM images, our purified kinetochores attached to microtubules. And so this is just one of the images that we obtained. You can see in this extremely large structure. It's greater than 100 nanometers. And the really exciting thing is we were able to see a ring encircling the microtubule that we believe is the DAM1 ring. And then there's other microtubule attachment sites. And now we're working with Ava Nogales' lab to really understand how these different complexes are binding to the microtubule. And of course, now we can do immuno-EM and other techniques to try to elucidate which proteins we're looking at in this structure and how they correlate with sort of the models that have been put forth for the kinetochore architecture. Next, I'm going to talk about the biophysical properties of the kinetochore where tension can stabilize attachments. So as I said in the first talk, tension is important to ensure that cells have made the proper attachments. So once kinetochores make bi-oriented attachments to microtubules from opposite poles, they come under tension and they're highly stable in vivo. In contrast, when cells make incorrect attachments that lack tension, there are error correction mechanisms that ensure that these are turned over to give the cell a chance to make the proper attachment again. When we started this work, the major mechanism known of error correction is via Aurora B phosphorylation. So the idea here is that the Aurora B kinase recognizes incorrect attachments. It then phosphorylates kinetochore proteins such as the NDC80 and DAM1 proteins that I told you at the beginning are major microtubule binding elements in the kinetochore. And that gives the cell a chance to make the correct attachment again. However, as I told you, our kinetochores don't have any Aurora B, so we weren't sure how they would behave under tension. So we decided to ask what the effect of tension on kinetochore-microtubule attachments is. And so the idea here is, you know that tension usually destabilizes attachments. And so the idea is, if we monitor the lifetime of the attachment at a range of different forces, how do the kinetochores behave? Now, for a typical protein-protein interaction where tension usually destabilizes it, the curve would look like this. As you increase the force, the lifetime of the attachment would decrease. And that's what we expected would happen because we were missing the Aurora B kinase on our kinetochores. So we at first looked at 2 piconewtons, and we found that the kinetochores would stay attached to the microtubule tips for about 20 minutes. And then the big surprise was as we increased the force, the lifetime increased. So by 5 piconewtons of force, the kinetochores were maintaining attachments for almost an hour. And this is actually longer than mitosis even takes in budding yeast. Now, as we continue to increase the force, the kinetochores started to just detach or rupture from the tip of the microtubule. So what we learned from this experiment is that something about high tension is really promoting attachments. And this is an intrinsic property of the kinetochore-microtubule interaction. There is no Aurora B kinase involved. Somehow tension is just stabilizing an attachment. And this is like those finger traps that you remember, as you pull on them, they come under more tension. And so we think that kinetochores have that property when they bind to microtubules. As we put them under tension, they are stabilized. This is reminiscent of two-state interactions such as receptors and ligands, where there's a strong and a weak state, and something about tension promotes the stronger state. And so we're actively working now on trying to understand the underlying mechanism where tension can really stabilize these attachments intrinsically. So in the future, we're using these purified kinetochores to continue to study the 3 major roles of kinetochores. The mechanical role, we're going to continue the EM work to try to really understand more about the structure of the kinetochore and how it physically attaches to the microtubule. I showed you today that we've confirmed that there's a ring structure at the kinetochore, and we're hoping to get higher resolution images of how the kinetochore otherwise is attaching. We're also probing the various functions of kinetochore proteins. We're going to continue to work on the biophysical properties. We discovered using these purified, reconstituted kinetochore-microtubule attachments that tension really directly stabilizes attachments, completely independently of Aurora B. So we're going to try to identify the proteins involved in that, and then how does that integrate with the Aurora B error correction pathway in the long term. And finally, I didn't have time to talk about it today, but we've been able to reconstitute major events of the spindle checkpoint using our purified kinetochores and identify some of the key phosphorylation events that recruit checkpoint proteins to the kinetochores. And in the future, we're developing fluorescent assays to look how tension and attachment affect the occupancy of checkpoint proteins, and we hope to eventually reconstitute the checkpoint in vitro to really understand the individual events that cause the kinetochore microtubule attachment defects to then halt the entire cell cycle. So thank you for your time, and I'd just like to acknowledge the people involved in this work. Bungo Akiyoshi pioneered the entire kinetochore purification. I've had help from a number of other people in the lab. Krishna Sarangapani and Andy Powers in Chip's lab did the biophysical work, and this has been a really long-term, fantastic collaboration with the Asbury lab. And Shane Gonin and Tamir Gonin were our EM collaborators who really helped us see the first structure of a kinetochore. And of course, I'm very grateful to the funding agencies over the years that have supported this work. Thank you.